Hello, my name is Alexander Orellana, and I am an undergraduate student at the Health and Equity Research Lab in the Department of Biology at San Francisco State University. And today I'll be presenting biological aging among Latina breast cancer survivors, also known as the Nuevo Amanecer 2 study. I would start off by saying that breast cancer actually is the most prominent form of cancer in women, and additionally, it has the highest mortality rate. However, in Latina breast cancer survivors, there's a large disparity. They experience higher levels of stress, anxiety, and overall a poor quality of life. But why is that? Well, a big part of this has to do with stress. Everybody undergoes stress, but when stress becomes chronic, it can weaken the immune system, facilitate the growth of these cancer tumors, and lead to premature aging. How does it lead to premature aging? Well, at the end of your chromosomes, we have something called telomeres, and they are a great biomarker for aging. As you get older, your telomere length shorten, but as one undergoes high levels of stress, this shortening accelerates. And additionally, there are many other factors that influence telomere lengths, as shown here. For a Noah 2 study, we have two primary objectives. The first is to characterize relative telomere length, and second, determine the effects of a peer-delivered stress management intervention on these relative telomere length. For our study design, we used a randomized control trial. 103 Latina breast cancer survivors were recruited, and all the samples were collected. This was known as baseline. After this, they were separated into two groups: standard of care and intervention. Standard care got the regular care, but intervention underwent a 6 to 10 week stress management intervention developed by Dr. Anna's group. And after the 6 months, their saliva samples were collected once more, and this was known as baseline. After collecting the saliva, we performed something called a DNA extraction and isolated the DNA. And we divided this to 5 nanograms per microliter in order to be used for the next step. And the next step, we actually used a fluorescent dye-based real-time qPCR using a reagent called CyberGreen. And we had two primer sets for this. The first one being for telomeres, and the second one for being our second set called the single copy gene hemoglobin. After this, a standard curve is generated after each run, and we look for three main things. The first is being an R-square range of 0.996 or above, an efficiency range between 90 and 110%, and a slope of -3.1 to -3.6. After this, we can actually determine relative telomere length using a TS ratio. But what is a TS ratio? Well, a TS ratio is the quantity of telomeres divided by the single copy gene hemoglobin. Over here in subject 1, we see a t-value of 37 and an s-value of 27, and we get a TS ratio of 1.37 when you divide them both. Over here in subject 2, we see someone with shorter telomere lengths. We see a t-value of 15 and an s-value of 23. Over here in this section, I'll talk about recruitment and retention. A total of 103 Latina breast cancer survivors agreed to participate in the study, and 101 provided samples at baseline, while 100 provided at follow-up. As for demographics, the average age of these Latinos were 56.1 years old, and many of them live in the United States for around 27.6 years. Additionally, 27% of them reported financial hardship, 78% had a high school education or below, while only 22 had above a high school education. And another factor we thought was the year since most of recent cancer diagnosis, which is 2.7 years. And all of these factors contribute to the telomere shortening. As for our results, at baseline, we see that our baseline TS ratio is 1.24, while our follow-up is around 1.26, which is not a large jump or anything. However, this shows that Latina breast cancer have short telomeres, especially when compared to a similar study. In the Broad et al. Study in 2017, in Hispanic women age 65, their telomere ratios are 1.4. However, ours are around 1.25, which indicates Latina breast cancer survivors have short telomere lengths. In conclusion, the Normal Institute has found a good way of collecting samples from this population, and we have found that relative telomere is a great biological measure for premature aging. Furthermore, understanding these roles of stress and premature cellular aging can provide insights into these molecular mechanisms driving these health disparities in these Latina breast cancer survivors, and find ways to help prevent these short telomeres. I would like to thank my lab and all the funding for supporting me during this time and allowing me to present. And thank you so much for listening.